and stuff with the ARCH samples. Then I'm going to get started doing those, and then I'm going to get the samples from Beaumont. So no, things are going very well.
Okay, and we'll get Zielski to help you whenever we get reopened again, if you need it.
Well, I think that that might be a problem because when we first open again, they're not going to allow two people in the same place.
Yeah, right. Well, I got other things for him to do. That's fine.
So, okay. I'm going to go ahead and mute myself again just to make sure.
That's fine. I'll mute too. There are 18 people listening in on us. Bye. Hi, Mike Petriello. Are you there? I could talk to you too. If you can unmute yourself.
Hey, Doug.
Hi, Mike. I just sent you an email about NIEHS has a new NOA or a RFA for COVID-19 work.
Yeah, is that the R21 or are they?
Well, I didn't read it actually. Dan Rapley just sent it, but I thought we could do the air pollution COVID-19. I don't know if we could do the mouse exposures yet. I think that's still BSLR3.
Well, yeah, I know Gil was talking about if it could be 2A or 2B, or is it still 3? I don't know.
I don't know.
Hey, Doug.
I asked Gil too.
Hey, Doug, I'm going to ask you guys to take it offline if you don't.
Okay.
Mute.
And Anna's going to start with just housekeeping. Yes. Yes, Anna, go ahead and start.
Okay, great. Thanks, Tracy. I was just quickly going to review some housekeeping items here. This may look familiar to the virtual meeting last week if you participated in that. For open discussions, just to note, everybody has been muted upon entry. When you want to talk, just make sure to unmute yourself, and then we ask that you go back on mute to minimize background distraction noise. After the presentations, there will be some time for Q&A. And we ask that if you submit a question through the chat box, that you start your question with the investigator's name that you're directing that question to, in order to help us stay organized and make sure we're addressing all appropriate questions. And then just some notes down there at the bottom if you have a spotty connection, if things look frozen, we recommend disconnecting from the VPN, turning off your video camera, or closing any additional internet browsers that you have open. And then quickly, this is just a screenshot of your view of Zoom and items that are of particular interest down at the bottom left, how to unmute or mute yourself. And then the chat box on Zoom, you hit the button at the bottom toolbar to open that. And then you can toggle between speaker view, gallery view, depending on your preferences there. But Tracy, I'll hand it back over to you.
Okay, so following our agenda, I'm just going to introduce someone you guys all know, who is Nigel Paneth, who's the communicating PI for the CHARM ECHO project. So Nigel.
Well, thanks Tracy and Anna, and welcome everyone. We have, in spite of the difficulties in having to do this online, we have apparently a very good attendance, and I'm happy to hear that and see that. And we hope to give you a good couple of hours. We can't give you lunch the way we normally do in these statewide investigator meetings, but we'll do our best to make it a useful 2 hours for you. By way of introduction, a few things. The COVID-19 epidemic has affected us, obviously. We can no longer recruit pregnant women. We can no longer do in-house, in-person assessments of children as we were doing. Children from the ARCH cohort, recruitment in the MARCH cohort. What we can do and have been doing is following up people by telephone interview. And in fact, the presence at home has actually made it a bit easier to access some of that information. So that's been going along quite well. And if there are questions about that, I think the staff can answer that very well, Alexa or others. I have been a little bit absent from the scene, and Jean Curver has been able to kind of take care of our study office for me. I've been involved in some COVID-related activities having to do with convalescent plasma that we've been working on. I'm hoping that I'll be able to come a little bit more back into the mix of things now. Things have sort of, our work, a lot of hard work has been done and there's more to be done, but perhaps the pace of it is a little bit slowed down. There are many important themes that you'll hear about today. One of the most important is which I'll talk about later in the meeting, is our attempt to create echo-wide papers. That is to say, not just papers that make use of our data, good as I think our data is, but really actually works together with other components of the echo world to write papers that have a larger scope, a larger sample size, perhaps more ideas. Several of those have already been proposed by our group, working with other parts of the echo universe, and there'll be more, I'm sure. And of course, I don't want to make it sound as though we're not interested in people analyzing our own data. You're going to hear some presentations that make use largely of our own data shortly. But the national scene, the people at the ECHO leadership is very invested in us having nationally a more robust representation of ECHO, what they call ECHO-wide papers, and we'll go over how those are created today too. But again, I'll say that's not to slight the work that's being done locally with our Arch and March cohorts, much of which can inform ECHO-wide papers. So that's all I wanted to say. Thank you very much for joining us. And I think the next person on the agenda is Kelly Herco. Is that right? Is Kelly on?
Hi, this is Kelly. Turn on my video.
Okay. I'll just say a word of introduction. Kelly Herco is a member of our epidemiology department, one of two members of our department who are headquartered in Traverse City. The other is Jean Curver. Let's say our Department of Epidemiology and Biostatistics. And Kelly was trained at the University of Michigan in epidemiology, also did a postdoc at Harvard, and we're very fortunate to have her in our department. She's interested in cancer epidemiology, but she's also taking a very important role in our ECHO project. So Kelly, please
Thank you.
tell us about your work.
Thank you for the introduction. So today I just wanted to talk about the review committee, which Nigel asked me to be a part of. And the purpose of this committee was to create a standardized process for collaborators to use the CHARM data, and also to try to assist the study staff to keep track of all of these study data uses so that we can report this and hopefully ensure further funding for the cohort. So what I'm going to present today is something that myself and Tracy Thompson and Doug Rudin have worked on as our kind of our first draft for how we see things going to create the standardized process. But I want to reiterate that, you know, this is going to be probably a work in progress, and we want to hear people's comments and questions, and we can certainly adjust our plan to meet the needs of the CHARM collaborators. What we've taken is kind of what has worked in other cohorts and tried to use those processes and tailor them for this cohort. But again, it is somewhat a work in progress. Next slide, please. So all of the information that I will provide the overview for today is found on the CHARM study website, and that website is listed there. If you go on the main website and then you just go on the For Researchers tab, you will find all of the CHARM study forms that we've put together. And basically, the CHARM study forms can be divided into those that are going to be used for investigators seeking to propose a new study within the CHARM data, or they can be used to submit an abstract or manuscript for review before submission to the journal or to the conference. And so for new proposals, we have existing data forms for researchers who are looking to utilize data that's already been collected and is already available. But we also have biospecimens forms specifically for studies which will be utilizing the biospecimens that have been collected. And then we also have an ancillary study form for researchers who are interested in some sort of recontact with CHARM participants to collect additional data for use. And then on the right-hand side of the screen, we have for the abstract and manuscripts, we've created a checklist. So these will help to ensure that the relevant grants have been cited, and just to clarify for anyone who will be submitting an abstract or manuscript that they are responsible for reporting that back to the CHARM study office so that for our data collection purposes, we can keep track of that information for reporting. Okay, next slide, please. So an overview here on this slide would be for somebody who is looking to start a new research project within the CHARM data. Our first recommended step would be to review the study website and to look at the other studies that are already in progress, make sure that there's nothing overlapping with what is already ongoing and supported. And then also review the code books and the data dictionaries so you have an understanding of what variables you would be requesting for your proposal. And then a list of biospecimens is also available for those interested in using those. And then you would fill out one of those CHARM study forms, so either the existing data, the biospecimens form, or the ancillary study form, and return it to the CHARM study office via this email. Eventually, we want the website to have everything be able to be submitted directly online, but it's not quite up there to that level yet. And so until then, we're just asking that the collaborators will fill out the form that they can download from the website and then submit it via email to the CHARM study office. And then initially, the review committee will screen the proposal forms to make sure that they're complete and also to make sure that they do not overlap with any of the existing proposals. And we'll also do our best to determine if something is feasible. So we may ask Tengfei to look into the distribution of the variables of interest to make sure that we have enough to fulfill the proposal that is being, the research that's being proposed. And then we will get back to the researcher within 2 weeks of the initial submission to tell them whether it's been rejected, approved, or approved pending certain revisions. And then once a proposal has been approved, we ask that investigators complete the data use agreement form, which I think has already been in use before we began these processes. So this is kind of the brief overview of the process, but all of the proposal guidelines are detailed on the webpage for those interested in reading more into it. Next slide, please. So just a note, I know I mentioned the ancillary study requests are going to be a little bit more intensive than an existing data request, because in these cases, researchers will be asking to recontact CHARM study participants. And so we just want to reiterate the fact that if this is something that is being considered, we ask that researchers really work closely with a CHARM investigator before submitting the proposal. And also just to note that any contacted participants will be made only by CHARM study staff, and so that should be considered for proposals when you're submitting for grants, that those funds will be needed to cover the time to recontact participants. And then also what we have added here is that any new data that's collected as part of these ancillary studies should be shared with the CHARM study staff. We will have, the applicant will have the priority to use the newly collected data within the first 2 years of data collection, but we do expect that the new data collected can be brought back for other research questions to be utilized in the future. Next slide, please. So for biospecimens, it's similar to the ancillary studies where we ask that collaborators work closely with the CHARM study office and try to give us some warning before these are being submitted so that we can obtain preliminary estimates for funds that will be necessary to access some of the requested specimens. And then also similar to the ancillary study request, we would like to have the information back to be used for other investigators after the proposed research questions have been answered by the researcher who submitted the request, but that after 6 months after the laboratory work, the original results should be entered back to the CHARM database for other CHARM investigators. Of course, after the researcher has used the specimens for their own research question, that would not be open to somebody else, but for other research questions in the future. Okay, and then the next slide, please. So for the abstracts and manuscripts under review, we had, I believe Tracy had sent out an email asking for volunteer reviewers. So thank you everyone who has volunteered already to be a part of the review team. And if there are others who are interested, please do email either myself or Tracy or the CHARM study email office address, and we will add you to the list. But what we've proposed is that even for abstracts, we want to have those submitted to the CHARM study office with the checklist. Again, the checklist is just going to make sure that all the relevant grants have been cited and the acknowledgement to all of the study sites is on there. And then once the abstract has been approved, we would like a copy of the poster and our presentation, again, for the records going forward. And then for the manuscripts, there's a separate manuscript checklist, which again, checks for the grant cited. And then for the manuscripts, we will be sending those to some of the CHARM reviewers to review and make sure that the conclusions are adequate and just an overall review, nothing too intense. We're not going to take too much of our reviewers' time, but just to give a little bit of an oversight to what's being put out from the cohort. And then once the manuscript is published, we will ask that the lead author provide the PDF copy of the approved manuscript back to the CHARM study office to again help with the tracking of the CHARM study data use. Okay, next slide, please. Okay, and then we just wanted to take a moment and have Tracy show the website just so you can see what I've talked about, where you can find these forms. And like I mentioned, eventually we'd like to have this all automated through the website so the forms can be accessed and submitted directly on the website. But until then, you will be able to find the forms on the website, but you will have to submit them via email to the CHARM study office. So Tracy, are you able to open up the website now? Not sure if this is working or not. If it works, great. If not, if everyone, when they are next on the website, want to take a look, and if you have questions, let us know. Again, if you go to the main CHARM page, oh, Tracy looks like she's getting there.
So can you hear me now?
Perfect. Yes.
Okay. So this is the front page of our website. Pretty simple, but the thing to point out is the tabs across the top. So you can see for researchers, if you click on that tab, the two things that are discussed for researchers are collaborations and funding opportunities. And so under collaborations, you'll go to the forms and proposal guidelines link, which brings you here. And you can see here that the guidelines are very detailed. But the main links are the existing data form, biospecimen form, ancillary study form, abstract and manuscript checklists. You can go and see the code books. So these are the code books that are freely available. Of course, we are in progress of creating other code books, so these other ones are not yet created to the point that they can be shared online, but we can get you some of that information. And then to go back, the list of studies in progress, these are not only the study's official aims that were created with the original application, but other things that are going on. So you can see the echo-wide analyses, the sub-studies that were started before ECHO funding, since ECHO funding, on and on. So this gives you an opportunity to see if what you're thinking about is already being addressed in some form. The last thing I would show you is the list of biospecimens collected or to be collected. So you would need to communicate with the study office or me to find out if these biospecimens that you're interested in are already collected or are to be collected or in process of being collected. But I wanted to list everything. So you can see here, so these are the March moms, and then the March child, same thing with the Arch cohort. So that's another place where you can get information. Is that everything you thought, Kelly?
I think so. Yes, thank you. That looks great.
Okay.
And we'll be probably making revisions as we go through the process. I think the last slide that I had really just said, you know, please let us know with questions or suggestions if you run into any issues, because again, this is our first attempt at creating these processes to make it work for this group. So we are definitely open to suggestions and questions. And that's all I have at this point.
Okay, thank you, Kelly.
Thank you.
Does anybody have any questions? We have a few minutes.
Yeah, can you hear me, Tracy?
Yes.
Yeah, this is Karen Rasico. My first question is something that I am kind of dealing with now with doing an echo-wide study, is the individual cohorts, just like us, have these committees, et cetera, that look at the manuscripts, et cetera. Some of them have like full committees that must review and give permission, for example, like with a research product, like a manuscript. And something we're dealing with is that the main ECHO office also has a committee, and they, like, when we're doing the ECHO-wide studies, you know, all those things are going to them. They have a committee, and they like provide the approval process. And something that we're kind of trying to figure out as we go is some of the individual cohorts, they also have very similar processes. So you're kind of, you end up, if you're doing ECHO-wide, doing this, like we have 4 cohorts, so we're trying to figure out how we don't have to have 4 separate review committees that might have different recommendations, et cetera, and so we're trying to make sure that we're not, you know, if individual cohort says, oh, we want this, but the main ECHO office says something else. And again, I guess this isn't really a question, but something that those guys and the DAC, they're kind of working through that now. So it's just something to just kind of think about for the ECHO-wide ones where, you know, is there a way where we can like do all of this once instead of doing it for each individual cohort? And then my one other question is, if people are interested in submitting a proposal, not through our local program, but say, you know, using the data or the research materials to put in, you know, an R01. Is there a separate, and I haven't looked through this yet, is there a separate process or something that you're thinking about where, you know, how we initiate that type of proposal?
Great. Thank you for both of those thoughts. I can take the first stab at it, but Tracy, Nigel, anyone else, feel free to pipe in as well. So for your first point, my thought would be that there's no need to do this if this is going through the ECHO-wide process. I think we kind of created these more for just papers that are going through just, our study group. But I can't necessarily make that decision. So I'll just say that's my preference for this. I don't think there's any reason to go through multiple different approval processes.
That's the way the others seem to be going. They seem to be saying, oh, if the ECHO main office says yes and it's going through that, they seem to be leaning that way.
Okay. Yeah, that would be my preference. First, definitely don't want to be creating extra work. And then for your second point, your second comment, now I'm blanking on what that one was. That was the
not like unsolicited, like if we want to not go
great,
through
great.
the ECHO program, but like if we want to submit an R01, like
Yes.
what would the process be?
So on our existing data form, for example, it says the purpose of this submission, and it says a grant or a paper or an abstract. So you could check just grant. If you're using just existing data, you would fill out that existing data form, and one of the questions will specify whether this is for a grant or for an abstract or a paper. And then that would be the same. So if you were going to be putting in a grant that required biospecimens, then you would fill out the biospecimen form, but click grant on that instead of the manuscript or abstract on there. So each of those proposal forms has a question that specifically reflects whether it's a grant application or whether it's for a paper or an abstract.
If I could weigh in, this is Nigel, just on one part of this. And we're very fortunate to have later in the meeting Beth Harris from the Coordinating Center to talk to us. But my understanding is that based on an ECHO white paper that I myself submitted a proposal, is that the approval processes from within the cohorts comes later, not at the initial stages. First, before I think they don't want, I don't think ECHO wants you to get all sorts of approvals before there's an agreement that this should go forward. So it's a matter of the timing of these approvals. Obviously, once you get 5 or 6 centers together, they'll each have to be willing to give this. But the initial effort, I believe, doesn't require you to first get the approval from each center. But Beth Harris, who may be here now or later, could perhaps respond to that.
She'll be joining at 2:30.
So we'll save that question for her. Make sure she answers that. When is the right time to go to get individual cohort approvals for an ECHO-wide concept paper?
Kelly, can I ask for some clarification regarding the ancillary studies and recontact of participants?
Sure.
And how that might interact with the local Michigan ECHO RFA? Because I'm putting one of those in this week, and we had originally proposed following the same workflow that I understood Sarah Comstock used, where the study office screened for the eligibility of participants, but then Sarah's team actually did the contact. With the guidelines you said for the ancillary studies, it sounds like that would no longer be possible. Is that correct?
If I could answer.
Yeah, that's probably better for Tracy.
I would just say that I think we put the wording so that you wouldn't assume that you would be contacting them, but I believe that would be, you would negotiate how the best way to do that with the study office. And Alexa Kruger may want to mention something, I don't know.
Yeah, I wondered if Rebecca would have this question because I know we discussed exactly that topic. So I think our thought is it probably is more study specific based on, but if you plan for your budget to have that you would do the contacting, we might just need to discuss that more, Tracy. I know we talked about things like Rebecca said, the opposite, and how Sarah's doing that and Rebecca would going forward, which affects budget and such.
I think we wanted to put the wording in to be a little more careful with the idea that you would negotiate it.
Okay, yep.
Yeah, I'd like to comment on this. It's very important, obviously, to coordinate these matters. And if you're going out to try to get resources from NIH to expand some aspect of our study, an add-on kind of module that does more communicating with participants, this has to really be carefully worked out budget-wise and making sure it's not interfering with anything and it fits. And so those steps have to be taken with our study office and our PIs.
Yes, and I certainly understand that if we're talking about an NIH grant application, we'd want to fund folks within the local team for the work they're doing. I was wondering whether for those local grant applications we needed to be budgeting for CHARM study staff and an already pretty limited.
Oh, local. I'm sorry. I thought you were referring to an overall NIH. Local. Well, if we haven't, I don't think, done that, but if there really is a burden on local staff, then you'd have to discuss that, with the study office. If you think there might be an additional set of tasks they have to undertake they wouldn't otherwise.
We have about 2 minutes left. Is there another question?
Hi, it's Kim Mickey. I had a quick question. Is this ready to be implemented now? Are these forms, I guess, ready to be submitted this week, or is there a timeline for this? And I also had a related question about ancillary studies. Are the folks who were part of those initial ancillary studies who would deposit and share data back with CHARM, would they be contacted for future proposals for the data that they would be sharing, or what would the process for those be?
That's a great point. I mean, with the first question, yes, the forms are ready to go and we're already reviewing things. So yes, they are live, but I do want to say as a caveat, if you notice things, please let us know, because it's kind of our first go around with it. And then with the ancillary studies, we did write in the detailed proposal guidelines that you can find on their website that we review the right to ask for the investigators to add people to that study who are CHARM investigators. And so that would be where a situation like this plays in, so that if you collected data and you wanted to say, I want to know who's proposing things and would be interested in being a co-investigator on all of these, then, certainly I think we'll have to work out that process exactly, but we do have it written in the guidelines for collaborators who are using the data that the CHARM study office reserves the right to request that specific investigators be added to this proposal. And so that would be a situation where that would play in, but we may need to have, for those ancillary studies, when the initial study has been completed, maybe a communication with the investigator to say, now, do you want to give the data over and you don't need to be involved with it at all anymore? Or would you be interested in being asked for your thoughts on any proposal that's coming through and being asked if you would like to be contributing to that? So that's a good point, that maybe we should add more of a formal back end process for those investigators who collected that data for their role to say if they would like to be included on future studies. So thank you, Tracy. We can talk about that.
On a related note, is there going to be any kind of boilerplate recommended language for the IRB consent forms indicating about sharing data back with the broader, with the original CHARM group and potentially with future investigators?
I don't believe we have that as part of our processes to date, but I can review, and I don't know, Tracy, if you have any thoughts offhand on that?
Only boilerplate we have right now is language for acknowledgements and things like that. But as far as consents, that's something we should look into. So thank you for that note. I am going to note that I'm sorry, go ahead.
No, finish off, Tracy, and then I'll
Oh, I was gonna try to move us on, so you finish your note.
Well, just the thought that if you do some, we've obviously established in our consent forms some sets of rules for returning information to participants and various other forms of what we can share. What we have, for example, rules now that we will not analyze drug data in the same pass as with identifiers, either ourselves or at the main study office. So we have these rules. So I think everybody who puts in some kind of a supplemental application, be it for local funding or for OIF funding or national funding, should just become familiar with what those sharing rules are and adhere to them. Now, if they want to, their own personal individual consents want to add or change other things, but they can't really move away too far from what we've already promised patients in our original consent forms, if those participants are being used. Does that make sense?
Yeah, I really brought it up because I'm also, I run the Turner Syndrome Research Registry, and we have a similar protocol where if researchers use us for recruitment and collect new data, that data is supposed to go back into the registry. So we've actually been working on having language to say to the investigators, put this in your consent form so your subjects know it's going to go back into here. So I was just wondering
Okay,
if
right. In other words, oh, okay, that's an additional feature, but I presume you also advise people that, you know, the original restrictions or promises you make in your original consent also have to be adhered to by anyone, you know, doing a secondary analysis or another form of data addition. Is that right?
Yeah, I think that's a great point, which I think is one of the reasons of having the prescribed language, because then everyone knows that they need to put that in.
I think we can
It's
build
a very
out
good point.
the consent.
Yeah.
I
We'll
see
work
those
on that.
points.
We'll try to put, I think it's a very good point, and we should make some comments on that. Yeah,
Okay.
we'll work towards that.
I am going to ask to move us on, if that's okay. So next up is Dr. Kerber giving presentations of current work and initiations, introducing that section. Thank you, Jean.
Well, thank you, Tracy. Hello, everyone. I just want to say it's so nice to be able to connect again with all of you, even if it's only virtually. It's not ideal, but we're all making do. And before we move on to sort of the next set of speakers, I just want to say, give a little credit to our study office. First, Nigel mentioned how well everyone's working. We have a great staff, a great set of people working in our study office. And as you sort of develop new research ideas, I just want to make sure everyone sort of considers them and thinks about how hard they've worked to sort of build this cohort, and maybe consider offering them a spot on, you know, some of your research products, if that should be appropriate. They might be too busy, and we would have to go through Alexa, who's our study office project manager. But just, I want to keep that in everybody's head. There's a lot of people working to build this, and we don't want to forget them as we move forward. But today, I have the pleasure of introducing our next three speakers that are going to talk about some of their charm work that's at different levels of completion at this point. So our first speaker is going to be Dr. Wan Ching Li Liu, who is an associate professor at Wayne State University. He's in the Department of Pharmaceutical Sciences there. And he's been at Wayne State for 2-3 years now, and we were thrilled to have him be the recipient of one of our local grant initiatives. Dr. Liu, I will turn it over to you now.
Hi, how are you? Hi, everybody. It's very good to meet everybody virtually. I know this is a hard time, but I hope everybody stay safe so far. Can you hear me very clearly over there?
Yes.
Okay, great. Yeah, so my project actually a collaborative one with Rita, Jean as well. They're both on board. So this project is focusing on a epigenetic regulation of polyunsaturated fatty acid metabolism pathway, placenta inflammation, and newborn health. Next slide, please. So I'm going to briefly introduce a little bit about the background regarding, the profile and the biology right here. So obviously here I listed, several molecules for saturated fatty acid on the top, MUFA, which is monounsaturated fatty acid, and PUFA, which is characterized with multiple double bond, in the carbon chain of the fatty acid. While in PUFA also, we kind of have a different, depends on the position of the double bond, it will be further characterized as omega-3, omega-6, or omega-9, or different other, classified groups. Palmitic acid, for example, this is the most abundant saturated fatty acid. Oleic acid is the most abundant MUFA, while linoleic acid, alpha linoleic acid are the most common PUFA in our diet and in our body as well. Next, please. So omega-3 and omega-6 PUFA, primarily we have to intake from our diet as a precursor. Some of the longer chain PUFA that can be obtained from, especially omega-6 group of PUFA, that can be directly obtained from meat that we intake. But majority of those PUFA, we do not make it at the precursor level on the top, LA, linoleic acid, or alpha linoleic acid, ALA. So those precursors are primarily in either nuts or fairly, chia seeds, flax seeds. Those are a little bit rarer, but you probably heard about it. So those precursors to get into our body will be further processed. And several key enzymes in the pathway are involved in this process, including fatty acid desaturase, delta-6, delta-5 desaturase. Those are the enzymes basically adding double bond between the carbon chains. And there are elongases. Elongases basically extend the carbon chains, from, you can see from the top 18 carbons extend to later on, 24, 25, 24 carbons. And obviously double bond getting increased as well during this process. One critical thing that I would like to point out is both omega-6 and omega-3 PUFAs play a very broad role in the biology, I would say. They're not only biomembrane integrins, but they also signal molecules that can be further processed, especially the long-chain PUFA like arachidonic acid, EPA, and DHA. So they can be further processed via multiple enzyme system to produce key molecules like prostaglandins, thromboxanes, leukotrienes. In general, omega-6 derived those type of lipid signal molecules are related to initiation of inflammation, while the omega-3 derived lipids are more likely involving depress or anti-inflammation. While EPA and DHA are primarily either we have to intake from the precursor and those are processed into a longer long-chain PUFA, or we can take from seafood like fish or fish oil. That those things we don't make in our body. Even the fish does not. They intake from the environment like algae. Next slide, please. So in the past million years, of course, you could see during the evolution, especially within the current centuries, we have underwent a huge difference in our diet, especially in the fat composition. We're taking more and more total fat. We're taking relatively decreasing the PUFA level in our diet. If you're just looking at omega-6 versus omega-3 PUFA, in our diet we get more and more omega-6 versus omega-3 in general. So in our ancestors that died, perhaps, we have a more balanced things, but now we back to like, accumulate to 20, even 30-to-1 ratio, omega-6, omega-3. So which has been blamed, for many of the biological issue and pathological issue and disease, for example, metabolism, chronic inflammation, obviously they might all related to those. Proof of course, for both the development of the fetus and also the health of the child are very important because majority of those things will be needed during the development, especially the early stage of the development, especially in the brain. So 40% of, if we take a fish oil, 40% of those DHA EPA will get into the brain. So the brain needs those very much. And a lack of those would definitely lead to a certain issue. Next slide, please. So there are many studies in the past that have been shown that imbalanced omega-3, omega-6 profile intake that is associated with all kind of health issues, as I mentioned earlier on, metabolic syndrome, cardiovascular disease, type 2 diabetes, hyperlipidemia, cognitive psychiatric disorders in children, for example, autism, depression, bipolar disorders are associated with imbalanced omega-3, omega-6 as well. So chronic inflammation, like asthma, chronic pain, they are also related to this as well. I think there are studies that showing that showing this figure that the lower the omega-3 profile, the higher the risk of preterm birth. Several other things related to the pregnant moms also correlate with this. Next slide, please.
1 minute, Dr. Liu.
Yes, okay. So the supplement of omega-3 will increase the health. Obviously, they could decrease the inflammation in placenta in this study, as shown in the right lower corner of this figure. Next slide, please. So we did some of the study that showing that some polymorphisms that may lead to proof of metabolism change. Obviously, in the fatty acid desaturase genes, the polymorphism lead to lower expression of FAS1 gene that may lead to a lower level of omega-3, omega-6 profile in our body. So the hypothesis came down from there, whether or not those polymorphisms will affect the proof of metabolism in placenta, so that it's going to affect the children's health later on as well. Next, please. And this is one of the studies that the polymorphism that I mentioned in fatty acid desaturase, that correlate with a clinical trial, give fish oil to the pregnant mom that prevent the asthma in their offspring, and that is related to the polymorphism genotype. Next, please. So the hypothesis of the study is basically we want to see leverage, the placenta samples that collected in CHARM, trying to see whether there's any epigenetic linkage between the proof of metabolism genes and the placenta inflammation, and then later on maybe the children's health. Next, please. So we select, the first image will be done by me, and this is genotyping whatever the placenta samples we have, and those are the candidate polymorphism that we selected from there. Next, please. And the next, AM2 will be done by Rita's lab. Hopefully we're going to sequencing some of the placenta samples so that we're going to connect all of those things together. Genotype, methylation, linked to gene expression profile, and the placenta inflammation, and later on child health. Next, please. Well, I mean, we've been talking a little bit about the MTA and later on about the COVID-19. I think we look forward to reopen the lab and finish the study as soon as possible. But we hope, by collecting some of the preliminary data, we will establish those genetic epigenetic connection so that we might deliver a potential preceding supplementation of fish oil in pregnant mom, and that maybe later on improve the health for child. I would thank, my collaborators and also Charm and Echo Polyphons. Thank the Wednesday Startup. Sorry, my overtime a little bit. Welcomes for some questions right there.
Yes, we have a few minutes. We have a couple of minutes for questions if anybody has something.
So the first question asking how are placenta samples standardized across subject? Yeah, that's a very good question. I guess, right now at this stage, we don't have, well, we had a meeting with Caroline in placenta analysis. I think, the question is still open. So we don't know what is the best way to looking at, but perhaps some of the histological level of inflammation of a placenta that, that we could look into it. And we could later on talk about some of the biomarkers like interleukin levels, TNF-alpha levels. Those could be some of the parameters that we look into it. It's still, in the exploratory stage.
Okay, we are almost out of time. Does anybody have a last question?
I mean,
Jean?
Yeah, Carolyn, yes, please go ahead and comment on the.
The alter ratio and health. So it's basically, you have a lower omega-3 lipids, and then that changed the balance about pro-inflammation and anti-inflammation balance. That was, blamed long for increase in the risk for, whatever the chronic disease. And of course, the polymorphism might linking this as well and making the things worse, because different people might have different level of the metabolism capacity that may further increase the risk.
Okay, well, thank you, Dr. Liu. In order to keep us on time, I guess I will move us along, but I would like everyone, if they have additional questions, to go ahead and type them in the chat. And,
sure, thank you.
You're welcome. Thank you. So you might get more questions as we go on, and we'll add those to the end. Dr. Salafia, I saw your comment that you would be happy to share more about placenta, but if we start that, it'll go a long time, I guess. So if we have time, again, at the end, we can talk more about that, but it is an ongoing discussion, as Dr. Liu mentioned. So our next speaker that I'm going to introduce is Dr. Rita Strakowski, who is currently an assistant professor in the Department of Food Science and Human Nutrition at Michigan State University. And Dr. Strakowski has been with MSU 2 or 3 years too. And she, like Dr. Liu, was one of the recipients of our local CHARM funds last year. And so she's going to give us an update on her project. So please go ahead, Rita. Thank you.
You're muted.
Okay, can you hear me?
Now we can.
Okay, perfect. So thank you, Jean. Before I get started, I really just want to thank everyone who's already spoken for the support for the small grants program. I also want to thank Ting Fei Ma for helping with getting us some preliminary data, and also to Sue Land, who's been really important for getting us samples and organizing things. So I really am grateful to both of them, especially. We're getting close to Mother's Day, and I'm talking about mitochondrial DNA, so it's pretty good timing. What I want to do today, we're also in the very early stages. We're just getting started. So I just want to give a little bit of background, similar to the first speaker, and a little bit of a plan going forward. Next slide, please. There we go. So this is our team. I'm really happy to work with everyone here. We have 2 of us at Michigan State and 2 at the University of Michigan, hopefully some familiar faces. And the 2 aims that I'll talk about today, really briefly, will take care of aim 1 here on campus at Michigan State. And then the second aim will be performed at University of Michigan, and then we'll kind of get together and coordinate data analyses. Next slide. Thank you. So our overarching goal, I'll start with that and kind of go into the background, do this a little bit backwards for the sake of time. As I mentioned, we're thinking about mitochondria, and we're thinking about specifically mitochondrial endpoints in pregnant women. And for this proposal, we wanted to look at differences in 2 specific mitochondrial endpoints in obese versus normal weight women. Can you move forward, please? Sorry about all of the animations. So our specific aims were to look, again, at 2 specific endpoints that are related to mitochondria, and specifically related to mitochondrial DNA. Forward, please. Oh, next. Thank you. And you can do the next. So first thing we're going to look at in aim 1 is mitochondrial DNA copy number, and I'll describe that in a little bit, as well as mitochondrial DNA methylation. And again, because we're using data, I had this on the front slide, but didn't mention it. We're using existing mitochondrial, we're using existing blood spots that have already been collected, and these were collected in 2 different ways: frozen, and they were frozen or stored at room temperature. So one of the important goals for us is also to do a little bit of methods development to try to understand whether one will be better than the other for these really important follow-up analyses. Next, please. So why focus on maternal obesity? Many of you may already sort of work in this field or think about this a lot. I definitely do. Pregnant women, more and more pregnant women are overweight or obese as they become pregnant. These data are from 2016, so it's a few years ago, but this is the prevalence of maternal obesity in the United States, and it's around 30%, 31%. Next, please. And what was important for us in thinking about this is that this is really similar to the prevalence in the ARCH cohort specifically, so the cohort we're working with. Next. And maternal obesity, in my mind, has consequences that are both short-term and long-term, both for the offspring as well as for the mother. Here, I just have some sort of thoughts that we usually have when we think about the dangers of having a woman who goes into a pregnancy being obese, especially at the higher end of the obesity spectrum. That includes early developmental milestones, as well as placental development or altered placental development, inflammation, toxicity, and then these very sort of long-term consequences for child health, and again, also for maternal health. And these are all short and long-term. Next, please. So why think about mitochondria? So those of us who are in nutrition and biochemistry, we often think about mitochondria as sort of the energy producer of the cell. That's the generic thought, but in reality, mitochondria within the cell has numerous functions, including dealing with inflammation, reactive oxygen species, regulating cell death, growth, adaptation, hormone production, and various other functions. So thinking about whether or not mitochondria are actually impacted in obese women during pregnancy is an important question. And it's actually not the first time this question is being asked. This has been investigated previously. Next slide, please. And the reason is really because what we think about in terms of obesity is that mitochondria is likely sort of in the center of both this upstream barrage of fatty acid overload and reactive oxygen species, and then that impacts mitochondrial biogenesis. And then it's upstream of additional reactive oxygen species production, decreased fat oxidation, decreased energy production, and then all of these long-term consequences for both maternal health as well as potentially child health. And that's where a lot of the work in pregnancy has sort of been over the last couple of years, is trying to understand how potentially a woman who is obese, how she passes on this message to her offspring. And mitochondria has been an important topic in that discussion. Next slide. So here are the two things that we wanted to look at. The first one is mitochondrial copy number, and I'll talk a little bit about this first. So there have been a lot of studies that have actually used mitochondrial copy number as a biomarker of mitochondrial function and potentially then also cellular health and phenotype and physiology and so on. So probably haven't seen a picture like this since, biochemistry in undergrad, but essentially, if you can, let's do next transition, please. Each cell essentially has numerous mitochondrial copy number, or numerous mitochondria, and each mitochondria have numerous DNA copies. And what researchers have done over the last decade or so is actually use really simple techniques, PCR, primers designed specifically to measure these, to look at how many copies of DNA each cell has or each sample has that's been isolated, and correlate that to various phenotypic disruptions and health outcomes, including obesity, and find that obese individuals or individuals with metabolic syndrome actually have disrupted mitochondrial copy number. Next slide. Similarly, AIM-2 of our proposal specifically focuses on DNA methylation. And of course, many of you have probably seen something like this, where we know that within the nucleus, DNA methylation has an important role for regulating gene expression, where cytosines are methylated or a methyl group is added to a cytosine, and this impacts the way that the histones may interact and the way that gene expression is actually, or the way that genes are actually transcribed. Next. But recently, probably in the last 5 to 10 years, we've also discovered that mitochondrial DNA is actually also methylated in a very similar way. The mechanisms are slightly different, but not that different. So what this is showing is, as I mentioned, so mitochondrial DNA is actually circular, not linear like nuclear. So this shows the 37 genes, and that's all that's transcribed from the mitochondrial DNA. And next transition. And right next to it is the bisulfite prior sequencing analysis that essentially shows areas within that DNA, or that circular DNA, that's actually methylated. And many studies have actually looked at this process in response to environmental exposures, specifically air pollution and various others. So one of the reasons we were curious about mitochondrial, or specifically looking at methylation, is that we've seen that nuclear DNA methylation is impacted in women who are obese, but no studies have actually looked at this in the mitochondria. And knowing how important mitochondria are for metabolic regulation and various other functions, this was an important question for us. Next slide. So just a very quick method, and should be finished. So we decided to use maternal blood spots as a source of genomic DNA for these analyses. This is just a picture off of the internet. It's not specifically the arched blood spots, but they look very similar. The study essentially collected blood spots from women at their first prenatal appointment, and half of each of the blood spot sheets went into the freezer, and the other half was stored at room temperature. So our goal is to first isolate DNA from these blood spots. Next, please. Next. And then what we'd like to do is use two types of technologies to look at. First, for AIM-1 mitochondrial DNA copy number, and this is simply going to use RT-PCR. And then for AIM2, we'll use bisulfite pyrosequencing. And again, the nice thing about these techniques is that they don't require us to actually isolate mitochondria. They'll utilize the DNA that we isolate from the blood spot and specific primers to be able to tell us, first of all, how many mitochondria the women have, or mitochondrial DNA copies the women have, and then also the level of methylation at specific genes within those DNA copies. Next slide. So just a little update about our progress. First of all, again, we're really early in the process. We, again, Sulan, thank you. All of the blood spot samples, it seems like, have been inventoried. And we're starting with 40 pilot samples that are ready for analysis and are actually already here at Michigan State. Thank you, Alexa, for driving through a snowstorm to get those to us. And then our U of M team already has a protocol for isolating DNA from blood spots, and what we'll do is confirm, again, whether or not frozen versus room temperature will work in our hands here. Next. So next steps for us, and this is really the rest of the project, is to quantify mitochondrial DNA copy number, first of all. First, we'll start with the pilot samples and then the full set of samples. And again, for the sake of time, I didn't describe the cohort very, very well, but we think we have access probably to around 400 mitochondrial, or 400 sets of blood spots. However, we'll be looking through those to make sure that the samples are good quality and that we can actually utilize what's there. So the final number of women included will probably differ from what we initially thought. And then once the first step is done, AIM2, we'll move on to University of Michigan to look at DNA methylation. And finally, as I mentioned, we'll get together and evaluate associations with maternal obesity. And very long-term, our goals is really, depending on what we find, is really to correlate these findings with birth outcomes, including things we know that are impacted in obese women, like gestational age of birth and birth weight, and try to start sort of understanding whether or not these mitochondrial endpoints are impacted in obese women, and whether that's potentially due to impacts on mitochondria. I think that might be it. Thank you, everyone. Again, thank you for everyone for all of their help and support and for getting samples to us. And I'm happy to take a few questions. And happy Mother's Day to those who are soon, to those who are moms and supporting dads. Thanks.
Just a minute or two for questions, and then you can enter them in chat if anybody has a question.
Anything yet?
Trying to give a minute. But if no questions right now, please do enter those in chat. And Rita, I love your shout out to Mother's Day. That's
yeah, it was just the time here. Last month would have been too early, so I'm glad this meeting moved.
Well, thank you very much, Rita. And don't go. We have time to come back for questions. But we will move on to try to keep in time. And so next we have Dr. Michael Petriello, also from Wayne State University in the Department of Pharmacology. Another person with links to nutrition, so this is just a very exciting day for me. But Dr. Petriello applied for one of the ECHO OIF grants, and so we are still hoping, fingers crossed, that he gets it. And I will just turn it over to you now, Mike.
No jinxing, please.
No.
Yeah. So thank you so much to all the organizers and Nigel for especially allowing me to submit that. It's been a really good way to meet all of you. But yeah, I'm going to talk about my work on impacts of environmental chemical exposures on gut microbiota and microbiota-derived metabolites in mothers and children. So you can go to the next slide. First, just like to thank everybody. I'm very new to Wayne State, so without Doug Rudin, he's got his nice little picture there. You're looking good there, Doug. Without him, I wouldn't even have known about the OIF or ECHO in general. So thank you. I have to thank Andrea Cassidy Bouchreau. We're very lucky to have her about, I don't know, a 5-minute walk from Wayne State. Excellent epidemiologist. Kim McKee, another co-investigator on the OIF. She's been amazing. She's an OIF awardee, I think last year. She does a lot of really good work. And then Sarah Comstock's also been a great asset. But a lot of my past research has been with environmental toxicology. So we've been funded a lot by the Superfund program. We have a P30 at Wayne. So bringing in all these different programmatic grants related around toxicology. Okay, you can go to the next slide. Just wanted to thank everybody. Okay, so today we're going to talk about 3 things pretty quickly, keep to the 2:15 time point. Since I am so new, I've only been in Michigan, I don't know, 6, 7, 8 months. So I'll give a little bit of background on what I've done in the past and hopefully where I see collaborations building. Talk a lot about pollutant exposures and microbiota disruption. It's not been a traditional target that a lot of people have looked at for pollutant toxicity. Briefly talk about the OIF submission, because at this point it's just submitted, so I don't have any data or results or anything like that. So we'll talk about the aims and that kind of overarching ideas. And then I have one slide at the end talking about future directions that I hope ECHO and our CHARM group and our Michigan group in general heads. And that's kind of incorporating environmental exposures and gut microbiota into other projects. Okay, you can go to the next slide. Okay, so just really briefly about me. I most recently was at University of Kentucky. I got to work with Dr. Andrew Morris. He's the guy on the bottom left there, who was the analytical instrument expert for the Superfund there at University of Kentucky. So I got to be able to work with biomarkers and targeted and some untargeted work. And it's a really good way to work with epidemiologists and people interested in the environment. So it's something that I've taken away with him. And I was also lucky that he was married to the head of cardiology at UK. So I was able to work a lot with some clinical samples in the atherosclerosis and cardiovascular realm. I've been really fortunate to be able to work with Dr. Sudha Bittinger, who works at Boston Children's Hospital. She's a endocrinologist, pediatric endocrinologist. And that's really where my kind of interest in pediatric and mother and children cohorts have come. Up until now, I've only worked in adult or elderly populations, looking at environmental exposures. Okay, next slide. Next slide. Okay. And now I'm really lucky. I've taken my K99R00 and I get to work in this beautiful building at Wayne State. The TMAO that you see there, is kind of one of the things that we're looking at with this OIF, but it's kind of just a proof of principle. TMAO is a metabolite that the microbiota can actually produce. And what's interesting about it, we'll talk a little bit about it later, it may actually not just be a biomarker of disease risk, but also a causative mediator. And that's one of the big things when you're always looking at clinical studies and doing association studies with biomarkers. Is it just a biomarker or is there actually mechanism? And that kind of came up with Wan Ching's slides with his omega-3 fatty acids. So that's where I am now. And now I'll go into a little bit about my research. Okay, next. So my bread and butter is looking at interactions between chemical and non-chemical stressors. And for the longest time, I've studied, I guess longest time, I'm still really young. I've studied diet as a combined exposure. I've looked at it both ways. I've looked at how diet can actually ameliorate or decrease the toxicity, but also how it can actually exacerbate it. And there's a lot of evidence out there, whether it's high-fat diets or trans fats or something like that, can exacerbate the toxicity of chemical exposures. But there's a lot of other non-chemical exposures out there. Think about right now what's happening, viral exposures. That's something really big as we can all start thinking about R21s and other RFAs. How do chemical and non-chemical stressors like that interact? Okay, next. Okay, so for those of you that aren't thinking about toxicology and environmental pollution all the time, it's a very broad kind of field, and because there's a lot of different man-made pollutants. There's tens of thousands of chemicals you come into interact with every single day. Here's just a few of them. And one thing that kind of brings all these into a big family or a tree is they're all lipophilic. So they all enjoy living in your fat. And that's what that middle picture is. That's adipose tissue. And one reason that I've studied these a lot is because you are primarily exposed through ingestion to these persistent organic pollutants. And because of that, and they also have very long half-lives. Because of that, once you're exposed, that doesn't mean that it's going to be excreted very quickly. So you build up a body burden over time. And so some of these are legacy pollutants like DDT, these polychlorine and biphenyls, some of these pesticides, dioxin-like pollutants. But on the bottom left and right of each of these are these emerging compounds, the fluorinated compounds, per and polyfluorinated compounds, chemicals, which are really big right now for Michigan and a lot of the country. And that's kind of where the focus of this OIF went. Okay, next slide. And to bring in the idea of microbiota, like I said, we ingest all of these pollutants. High-fat foods, dairy, meat, some fish are highly exposed to these. So that's how we get them. And so what is the first place for a target of toxicity? It could be your gut. And not only that, a lot of these pollutants, because they have long half-lives, are circulated throughout the enterohepatic circulation for a long time. So the gut microbiota may come into contact over time. Next slide. So my background is primarily in preclinical models. So using mouse models to study whether it's atherosclerosis, cardiovascular disease, diabetes, that kind of idea. But we've gotten interested in how pollutants could also modulate the gut microbiota. So just one or two slides on some of this past work. We actually developed a mouse model of a dioxin-like pollutant exposure using dioxins as a model for toxicology. And we fed them special diets that they weren't going to get fat, but they would still show the endpoints that we were interested in. And we can talk about why we chose that in the chat. But we can go to the next slide just to show some of the data. We were very interested in seeing how these dioxin-like pollutants could modulate gut microbiota. And so we did 16S ribosomal nucleic RNA sequencing. And these are some of the things that you get whenever you see a gut microbiota 16S sequencing paper. You can look at changes at the phyla. We did see significant differences due to PCBs. You can look at alpha diversity changes. You can look at beta diversity. And then you can drill down into specific genres and even species if you do really good sequencing. And that's exactly what we saw. The exposures in the mice led to changes. You can say bifidobacterium, lactobacillus, some of these bacterial genres that are actually added to yogurts as like a protective kind of thing. So it's modulating the gut microbiota. So next. And so you can do 16S sequencing, but another thing that we were really interested in was looking at metabolites that the gut microbiota produce as another way to kind of show that the pollutants were changing or modulating the gut microbiota. And so TMAO is cool because it's dependent on what you eat, your gut microbiota and your endogenous enzymes. So say you ate a big steak, your gut microbes can turn the L-carnitine into that into trimethylamine. TMA is very stinky, so probably not because of this reason, but it's lucky we have these enzymes that can turn TMA to TMAO. And then this TMAO, this trimethylamine oxide, actually circulates as a biomarker and may actually be a causative mediator of disease too. So next. So we wanted to look at how microbiota can change during pregnancy. And what's the most important thing for this slide is in the blue here. The maternal gut microbiota shifts during pregnancy. And so if we know that environmental factors can change the gut microbiota, maybe they can actually screw up this shift. And maybe that can actually lead to adverse offspring outcomes. So that's kind of the background for the OIF. One more slide, please. And so there's only one paper, if you do a PubMed search, there's only one paper out there on, in humans, impacts of POPs on maternal gut health. It's actually a really good paper if you're ever interested. Here's, you can just copy this, the title. But they were looking at how changes in pollutant concentrations in the breast milk were associated with changes at the gut microbiota level. For the OIF, if we click one more, we're really interested in how maternal levels in third trimester individuals can lead to changes at the gut microbiota level. So OIF application overarching hypothesis is that maternal exposures to chemical pollutants, we focused on PFAS just for Michigan, but it can be related to other pollutants, leads to decreased microbiota diversity and taxa-specific changes, which is vertically transmitted to the offspring. And these changes in maternal gut microbiota lead to dysregulated production of microbe-derived metabolites. And so one thing with the OIF, you really want to leverage more than one cohort. So we're lucky to work with people with the MAP and the MARCH cohorts. And what makes these really unique is they have collected maternal serum and feces samples. And then also there's infant fecal samples, so we can look at longitudinal changes. Okay, next. Specific aim ones to test the hypothesis that maternal exposures to the PFAS, again, we're focusing on PFAS as a model, associates with modulation of maternal gut microbiota and the metabolites, and with decreased infant birth weight. That's the outcome that we're interested here. And the second aim to test the hypothesis that maternal exposures predispose their children to suboptimal microbiota structure and function, which is conserved up to 18 months. So we're lucky that they've taken the fecal samples in the children longitudinally. So that's something really interesting about those studies. And one other exciting detail, we're going to use metagenomics approaches. So if you do 16S sequencing, it's really hard to get anything better than genre level. With metagenomics, you can actually look at strain changes. So we can actually see what strains are being passed from the mom to the babies. Okay, one more. And then, so this is the last slide, just generally environmental chemical stressors and charm. Currently, it is a major focus of the AIM 1. People are using dried blood spots to look at POPs and heavy metals over 2 generations, which is really interesting. Very focused targeted analyses. And then environmental pollutants as mediators of the outcomes of other aims. That's really the big question. So it's a focus of AIM 1, but can environmental pollutant exposures be added to other aims of charm currently as mediators or modulators? But in the future, I think where charm should go, perhaps, and other echo groups, they're going to look at more untargeted analyses. There's a term called exposomics, and then it kind of goes hand in hand with metabolomics. How can we look at a broad range of exposures with relatively small sample size to get a better idea of total body burden? I think that's something we should start thinking about. But obviously there's pros and cons, and we can chat about those too. And then how can we leverage this resource that's available to all of us to hear, or should we use internal options? So with that, I'm all done. Thank you again. Take any questions.
Yeah, thanks very much, Michael. Very interesting. Do we have any questions for Michael? Time for about one question, really. I see nothing in the chat box. So thanks, Michael. We'll just move on then to the next part of the program. You've heard a lot. Someone just came in. Oh, no, it's for Dr. Liu. Okay. We've emphasized in these 3 presentations the way we use our local database, and now we're going to talk about increasing what we can do to increase our echo-wide concepts. And we have, in order to do that, we want to make you acquainted with the national scene, the national location where you can find out about all the things you need to know in order to develop an echo-wide concept. So first, Tracy's going to talk about the Metadata Catalog, which exists on on the national website. And then Beth Harris has, of the coordinating centers, is kindly agreed to come to us and discuss the ECHO process for creating ECHO white papers. So I'll start with Tracy.
Hi. So I'm going to share my screen to make sure that I can show you. Basically, I wanted to make sure that all of the people in the CHARM group would know that these resources were available. So this is the ECHO-wide on the national website. And this is one that you do have to get access to. And I'm sorry, any question, Nigel? No?
No.
Okay, so for those of you who already have access, that's great. For those of you who may not yet have access, you can reach out to me and I can help direct you that way. They do have a process for doing that. So there are 3 different websites. There's the SharePoint, the Learning Management System, and the ECHO portal. The SharePoint is really a website for announcements and information and those things. What I'm going to focus on today is the ECHO portal. So when you enter the portal, you use your login, and this is what you would see. I hope everybody's seeing my screen okay. So there is a lot of information here, administrative reports, different libraries of surveys and other things. There's the BioTrack, which is used to track the biospecimens that will be going to ECHO. I'm going to focus on the Metadata Catalog, which is this one over here. So you simply click on, and you'll see that this is a catalog that ECHO has created with information on all of the different cohorts and awardees. And so just their way of, their language is that an awardee is like our group of 5 organizations and 6 PIs in our award from ECHO. And so we are a single awardee. However, we have 2 cohorts, Arch and March. So if you're looking for cohorts, or you could look for awardees. And so if you go into cohorts, this is a list of all of the cohorts. And of course, there are 86 entries. So this is where, if you are interested in finding out if other cohorts have samples or data that you could either use for a potential manuscript or an ECHO-wide concept, you can go into this database and you can find things that you may be interested in. For example, if you wanted to see who has existing blood, whole blood, so you can click on that and it will pull up 29 entries who say they have existing maternal blood. You can also ask who has assays that were already run on, let's say, folate. So there are 5 entries of cohorts that have already done, they have existing assays already done on folate in the mother. So you can go through all of these different categories and search depending on what you're interested in. And so one more quick example I'll show you is, so for example, if you're interested in the child's built environment, so do we have any activity related information? Okay, that wasn't a good one. That was because nobody has that. But anthropomorphics, so do we have birth weight on the child? Oh, that can't be right. I must have, I know what I didn't clear the list. Yeah, let me get rid of this one. Sorry about that. Okay. And so in other words, this is all to show that you can find those cohorts that have the things that you as an investigator are interested in potentially looking into. And as you saw, I accidentally had multiple things. So you can look for, do we have the mom's blood and the baby's birth weight? And so those, you can look for multiple things at one time. So this is just to hopefully show you guys some of the opportunities. And I'm going to ask Anna if you will go back to the slides and let me unshare. Okay. Thank you, Anna. So I do want to see if Beth Harris is on yet.
Tracy, this is Anna. She did just join.
Okay. I just quickly will ask if there are any really quick questions on the echo-wide metadata catalog that I just went through with you. Okay. Anna, will you please introduce Beth and make sure I give her the that, because Beth is going to oversee talking to you guys about the ECHO process for ECHO-wide papers and concepts.
Sure, yep. So Beth Harris is a project leader at the ECHO Coordinating Center, and she works on the publications committee. Beth, can you hear me? You may be on mute.
I sure can. Can you hear me?
Yes, we can.
Perfect.
Thank you.
Okay, so I'm gonna just jump right in to SharePoint. So I will share my screen. Okay, hey, yes. And share. Okay. So first of all, I do need to ask for everyone, in order for you to use SharePoint, you need to have access. And if you do not have access to SharePoint, into the ECHO SharePoint site, please let Anna or me know, and we can get you the help that you need going through getting the proper password and everything. So you have to come through the Duke system first. Anyway, so just please send us your names and we'll get you sorted out. So the PUBS program is here on, of course, the ECHO SharePoint. And actually, I'm going to start my video so you can at least see my face. Okay. So here's the ECHO SharePoint, and you can get to the publication sites in a couple of different ways. Here you can click on the PUBS program here, or you can come up here to the publication tab up on top. And I think you guys might have gone with the overview that the publication process is a 7-step process. So here are all the different 7 steps in the process. But for the most part, what you will use SharePoint for is step 1 and step 2. And so I'm going to enter through the blue button on the main pub page. And when you go there, this will pop up. And please interrupt me if you have questions, and I can try and help clarify. I know I have a limited time to go through this. And I do want to offer, after this presentation, if you're still not sure, reach out to me and I will walk you through this process again, and how to submit a concept and a proposal. So here on the main page, once again, is I sent you guys all this flow chart that it's even, I think I sent you one that was even more detailed in terms of what the process is. But for today, you're going to go into step 1 to see and launch analysis concepts. So you guys might have received emails stating, hey, there's a new analysis concept, go check it out, go respond to the discussion board. You might have seen it in the ECHO newsletter saying, here are all the analysis concepts, go read them and comment to the discussion boards. What that means is that you're going to come here into step 1, and you're going to click on the green button to look at the open analysis concepts. And this is where this is at, where everyone has listed, these are the 4 recent open analysis concepts. And these concepts are just ideas, and they're put out by anybody in the ECHO community that they want to either get people involved on the writing team, or if they're requesting biospecimens, they're just asking people to comment on what they're planning on doing with ECHO. For an ECHO, either for gathering ECHO data or what their idea is and how it could benefit ECHO. And it's just it's a peer review type deal where it's just bouncing off, trying to get people to bounce ideas off each other. So if we go into the first one, this one is by Andrew Desmukes. You click on it and it will give you the very brief idea, who it is, the hypothesis, objectives, and some other key data that we just keep just for behind the scenes of like, do you want to use biospecimens, yes or no? And then this is where you would actually respond. So after you read it, if you so inclined that you wanted to join the writing team, you would just respond saying, hi, I would like to, respond, join the writing team. And at the end of these discussion boards are open for 2 weeks, at least 2 weeks. The writing team leader will get these replies and then who has responded to their discussion board. But I just wanted to stress that's what we try and promote in our newsletter, people coming and looking at the discussion boards. So, but in order for you to actually submit a concept, you would come to this page and you hit submit a new analysis concept. And what it does is that it pops out an online form, and this is what we use to create the discussion board. And once again, it's the, just some details as far as the writing team leader, phone number, email, who you think you're going to have on your writing team. This is all going to be ECHO-wide, and it's all going to be on the data portal. Hypothesis, objectives, and just to get some general ideas. And then when you're done, you submit it, and it gets sent to the pub's mailbox, and then the operational team will get it up onto we'll open the discussion board. So your discussion board is open for 2 weeks. After 2 weeks, we will close it, and you will receive whoever has responded to your discussion board. And in that time, if you're asking for biospecimens, DAC will have here will have contacted you if you're looking for biospecimens, and also at the same time, DAC will have contacted you or will be in some way to let you know that they're going to assist you with your data analysis plan and try and work on feasibility that you will need to know in order to progress it onto an analysis proposal. But here on this page, I also wanted to show, if you are curious to see what other analysis concepts have been submitted, the closed ones are all the discussion boards, the analysis concepts that have been since the existence of ECHO, and you just toggle through, and by clicking on the discussion board, you could read what the description is. I'm going to go back a page. And my internet's frozen. Oh, come on. Okay. You could see the analysis concepts that have moved on to analysis proposals. So this is a list of writing teams that have already started and have gone through the process into step 2, the next step. And I'll go back a page. And there's even one that we have where people have retired or withdrawn analysis concepts. But this is just a tool to get involvement from the ECHO community, to see what everyone else is doing. So when you're ready, after your discussion board has closed, and you're ready to submit an analysis proposal, you click on step 2. Step 2 will show you your proposals. And when you submit a new proposal, you click new item right here. And you're seeing the ones that I have added into the system. Here's Nigel's. I helped it, Nigel. Further down the page, there's proposals under review. So these are all the proposals that are currently have either been prepared for the steering committee approval, which this one has, or Same with this one. This one here is actually is in the process of being reviewed by the PC. And then further down, you can see all the approved proposals, and you can click on there to actually read the proposals and see what their manuscript is ultimately going to be about. So in order to submit a new proposal, you click on New Item, and it's an online form. And I'm going to say right now, this is not perfect, and we're trying to update it as we go. It's a little clunky, as with SharePoint. All I can say is if save often, which is here is the save button. It's a little clunky, but we have a new software that we're trying to implement behind the scenes to make this more user-friendly for form making. And so that's one of our future improvements, but we're not there yet. But this form just gives us like the general information. First question is what is your analysis concept? So here are all the analysis concepts. So what you would do is you would find your analysis concept that just closed, select it, and I'll just click that one. You enter in your text, your name, your writing team members, and it's just a various of other information, either, contact information. And then when you're ready, you hit next. Oh, and it's not letting me because I haven't done something. Oh. So when I hit next, it will proceed you through more questions. And if any space and time that you guys have any problems or questions, contact me at the ECHO program through the ECHO CC mailbox, and or not the ECHO CC mailbox, the publication mailbox, and I'll make sure Anna has the email address, and she can make sure that's all sent out to you guys. So feel free to contact me, and I will help you guys as much as I can. And so you fill it out, and then ultimately there is you're going to end up filling out. I think if it will let me, I might have to trigger something. Let me see. I don't remember. Oh, there's an ECHO COI. It will ask you. We have questions about ECHO COI and all this other fun stuff that we want to make sure people do. Okay. Anyway, then the last page is mostly for to explain how to what is your proposal, and it's the background, the hypothesis, specific aims, and you would fill this out. So knowing that this form is difficult, there is also a paper form that you can fill out, a Word document. So I'm going to close this. And the access to the paper form is in this last tab, which is the toolbox. Here is the analysis proposal template. And if you click on this is just the Word form of this. This is an alternative that after you put in your basic information into the form, if you want to come over here and just fill out the last part, like from what page number is this? Anyway, type section D all the way down to you start describing your paper, and I know I'm probably making people dizzy. You can use this, save this, and send this. You can attach it in the Word, in the online form. So if I go back, you could fill out this form, save it to your desktop, come back to the analysis proposals, hit a new item, and there's an attachment function. Click here to attach a file. If that works for you, that's fine too. Like I said, we're trying to improve upon this. It's clunky, and we're trying our best, but we got to use with what we have. Then once you guys have submitted these proposals, we get an email in our mailbox. We then progress it through the PC committee. So the PC committee, and I think maybe, I don't know if you went over it at all, but it consists of an associate chair and 3 primary reviewers. It usually takes about 10 business days in order to get a review back. Once you get the review back, you can you'll have a decision of whether or not it's approved with revisions, or needs revising, or not approved. and that you can take the changes that are provided, or the suggestions that are given to you by the publication committee. You can revise your proposal and then submit it back to the publication mailbox for it to be sent to the steering committee approval. Now, steering committee approval takes place with the steering committee meeting, which is the third Friday of every month. And just to let you guys know, like throughout this whole process, you will get emails from the pub's operation group to make sure you know where you are in the step. You know, every time we return something to you, we tell you what the next steps are and what you need to do in order to get to the final approval from the steering committee. And then once you get approval from the steering committee, you will either, you're either going to go to step 3, which is analyzing data, which is you work either with the HERE to get your data through biospecimens and running assays, or you will work with DAC, and DAC will work with you to work up your analytical plan and provide the final results to you. Step 4 is to draft the research products. And research products is anything, abstract, presentation, or and a manuscript. And it kind of gives you the whole thing where once you submit an abstract or a manuscript, once again, it gets reviewed by the PC, and we try and keep the same reviewers. So there's a continuity of the reviewers from the very beginning, from the analysis proposal, all the way to the final manuscript. And then you just, once you get reviews back, you can either, revise and resubmit. And then finally, you have a product. We offer editorial services for the final manuscript going to the journal. But in general, we're here to help you. And I know this was rather quick, and I only have a few minutes left. But if anyone has any questions, please let me know, and I can try to answer them.
Thank you so much, Beth. That was a really wonderful overview. I mean that, and I can affirm from personal experience that Beth and Anna have been really, really helpful in when I was trying to get my concept moving, and any difficulty I ran into, Beth was always available to dig me out of the hole that I did. And you've also heard, investigators, 2 separate offers to help you get on the ECHO websites, first from Tracy and then from Beth and Anna. So if you have any difficulty, a lot of people can help you get onto the ECHO website so you can see all the things that are going on, the metadata catalog and so forth. It's
Nigel, could I add one thing?
Yes, please.
I just wanted to let everybody know that this has been recorded. It's being recorded, and we will send you out the slides as well as the link to the recording. And in the slides is a presentation that CC had created that takes you through what Beth has taken you through. So there will be some slides to help you figure it out if you need to go through that. Thank you.
Yeah, and that slide set is a little outdated, but the overall arching idea is the same.
So before we let Beth go, I'll just ask if there are any questions for her. Anybody wants to ask? Okay, not at the moment, but I'm very, I think Beth will be pleased to get your queries in other forms by email or whatever. And we do so appreciate you taking the time to come and speak to our group today, Beth. It's really very special to have you.
You're welcome.
Really, I mean it.
And I'll put my email as well as the pub's mailbox in the chat before I log off.
Okay, that's great.
Okay, thank you.
Thank you, Beth. Well, it's been given to me to talk a little bit about, to summarize where we are. And I think what you've seen is that we have, I'm astounded that we managed to get no fewer than 60 or 62 or 63 people to join in this virtual meeting. which had it been in person, that would have been a record for us. I think we've had 50 before, but maybe not 60.
Our highest number today was 72.
Oh, really? Oh, okay. That's amazing. And it testifies to the talent pool we tap into in our 5 institutions in Michigan, the 3 large universities, Michigan State, University of Michigan, Wayne State University, plus the Henry Ford Health System and the Michigan Department of Health and Human Services. And my view is that Charm Coalition is trying to make our 2 cohorts, Arch and March, in which we are continuing to collect data. Arch is a closed cohort we're doing follow-up on. March is still an open cohort. We're registering new patients all the time. And make that basically a platform for investigators across our state to say, how do I take my ideas, perhaps my ideas have been formed around environmental issues, perhaps around nutritional issues, perhaps around many other issues, but how can I make them link to this resource, which allows us to essentially be driving forces in improving maternal and child health, not just to write papers, but to actually improve health. And I think that's a feasible goal. And you've heard both the opportunities for local work that are exemplified by our 3 presenters who talked about the grants that they got from our coalition, not from the NIH money, but from our VPs for research of our institutions to support young investigators coming in and doing pilot work with us. And you also heard about how you could move it up to the national level or with collaborators across the country. So many opportunities to really thrive and grow your careers using our study as a place to start and to move forward. So I want to thank everybody for coming here, and we're always open to questions. As you know, we have no fewer than 6 principal investigators. I'm just the contact principal investigators. We have 2 at Michigan State, Gene Curver and myself. We have Doug Rudin at Wayne State, Michael Elliott at the University of Michigan, Charles Barone at the Henry Ford Health System, and Patty McCain at the Michigan Department of Health and Human Services. So we have a distributed structure, and everybody's really eager to make as many opportunities as possible for investigators who would like to join in this effort. And with that, I think I'm going to invite any of my fellow co-investigators to make any statements they'd like to make, Gene or Doug or anyone. Okay. And is someone is, yes, Rena Ficharova.
Yes.
Good to see you here.
Your adopted child from Boston is here.
Yes.
Just wanted to say hello to everybody and to congratulate you on this amazing achievement that I heard about today on all fronts, these fascinating projects and collaborations. And thank you so much for making me part of your family. I think there is so much to look forward to. And this world belongs to science. Yes.
Yes, it's for science. I do want to say that to let everybody know that we have 2 very, very important collaborators who are outside the state of Michigan. One of them just spoke to you, Rena Ficharova, a pregnancy immunologist and a wonderful collaborator at the Brigham and Women's Hospital and Harvard Medical School. And also, she hasn't spoken up yet, but maybe she'd like to, Carrie Salafia, our extraordinary placental pathologist in New York, and who has been the custodian of many hundreds of Michigan placentas, flown over to her wonderful laboratory to do all sorts of innovative placental studies. And just invite, Carrie, do you want to say a word or two? I'm not sure Carrie's still on. I know she was on earlier. apparently not on. And I might ask Susan Lesig. Susan, if you're on, would you like to say a few words? Susan is our project officer at NIH, and I think for Susan to see how our whole collaboration works together, do you have anything you'd like to suggest or comment, Susan?
Hi, Nigel. Yes, I was able to get on and listen to some of the presentations of the current work going on, and it's very, very interesting and exciting work. I don't have anything specific to say, but thank you for inviting me to listen in to the work that you're doing in Michigan. So
well, we very much appreciate the support we get from our colleagues at NIH, and as well as the other components. You've heard from Beth Harris from the Coordinating Center. There are many components to this project. I should thank them all. The Data Analysis Center, DAC, under Lisa Jacobson's great guidance, as well as the program office that Susan represents. And it's quite a large and complex ocean-going liner, and we're just probably one of the lifeboats at best. But it's fun to join in, and we welcome everybody who came to this meeting who would like to make use of our data. Okay, so thanks very much. Is any final comments anybody wishes to make? All right, thank you. Stay safe, stay healthy, be well. Good to have you with us today. Thank you.
Thank you.
Thank you, Nigel.
Cheers. Thanks, everyone.
Thank you all.